Welcome to Lecture 3 on the topic of nutrient uptake. This lecture is part of plant physiology subject, which is a component of the Bachelor of Agriculture and Technology, a degree offered by both Melbourne Polytechnic and Lateral University. Please visit our website at www.melbournepolytechnic.edu.au for more information on this course and other courses that we offer. My name is Dr. Nikki Cooley. We are learning various aspects about plant physiology by taking a virtual journey through the plant. By this stage you will have watched Lecture 1, the introduction, followed by Lecture 2 on root structure and function. This then brings you to this lecture, Lecture 3 on nutrient uptake. When you are considering nutrient uptake, you need to be mindful that this is in the root environment. So some of the concepts that we touched on in Lecture 2 in the root structure and function will be relevant here too. In this lecture we will be looking at the functional role of nutrients. I will be giving you an introduction to nutrients in the soil environment. We will then be looking at some specific aspects of nutrient uptake. This will include root structure and function, physical and chemical attributes of uptake, and finally nutrient uptake pathways and associated mechanisms. In Lecture 2, Part 1, we explored the role of water and why it was important for a plant to not only efficiently take up this solute, but also that it needs to be selective in this uptake. Well, it is no surprise to you that nutrients also play a critical role in many of the plant's functional roles, which impact on all aspects of plant growth and development. In the second stage of this subject we will be exploring in detail the many functional roles of nutrients in plants. In fact, without nutrients, plants are unable to complete their life cycle. Inefficient nutrient availability or uptake as well as excess uptake of nutrients can result in significant impacts on plant growth and development. Water and nutrient uptake are integrated. It is important to understand that often adequate water in the root medium is required to uptake nutrients effectively. In this lecture, we will concentrate assimilation of mineral nutrients on higher plants. Higher plants are also called autotrophs, and they can synthesize all their organic molecules out of inorganic nutrients. This will involve firstly the absorption from the soil, and secondly incorporation into organic compounds which are essential for plant growth. This incorporation of mineral nutrients into substances such as pigments, enzyme cofactors, lipids, nucleic acids and amino acids, for example, is known as nutrient assimilation. Nutrient assimilation can be defined as the conversion of nutrients into fluid or solid substances in the plant. The processes of some nutrients such as nitrogen and sulfur is among the most energy-consuming reactions in all living organisms. In order for nutrients to be assimilated into the plant, they must follow a pathway. The following slide illustrates 6 essential components of this pathway. First of all, roots absorb minerals, water, oxygen and toxins. These minerals are then translocated, which means transported, through the plant via the organ, the xylem. It is the process of transpiration, which we cover in another lecture, the loss of water from leaves, that enables the movement of minerals through the plant. Minerals are then used in several processes, from cellular respiration all the way through to photosynthesis. The byproducts of these components are sugars. These sugars can be then used as stored and translocated or moved around the plant via the phloem, where they are stored back in the roots. It is important to note that not only do you need good water uptake to take up minerals into your plant, but you also need good oxygen. This is often missed. So let us look at some solute transport basic facts. When the stomata organs are open, the plant is transpiring. Solutes can move from the soil into the root. Solutes are transported to the centre of the root in a structure called the stele, where they reach the vascular bundle. In the vascular bundle, they can either enter the xylem or the phloem, depending on the physical chemical properties of that solute. The Casparian strip is a component of the cell wall outside the stele, but within the root structure, and it is important as it prevents the loss of solutes from the root. The processes results in the roots being under tension. Therefore, water potential plays a key role in plant solute uptake. In order to truly understand how nutrients are assimilated into the root and then the plant, you have to understand or be familiar with the root structure. As part of your DIY practical, you will need to examine some roots under the microscope and label their structures. This will enable your understanding. So nutrients will enter the soil solution, they will travel through the epidermis and cortex, through the endodermis where the capsine strip is located, and then into the xylem parenchyma. Some important facts to take on board. Nutrients will only enter the root at the root hair. The structure and the architecture of the root can alter the rate of nutrient uptake. Nutrient uptake is similar to water uptake. The nutrient is taken up at the root hair. It then needs to travel to the phloem. There are several pathways or journeys it may take, and this depends on the chemical and the physical components of the nutrient. The images on the slide show the rough pathway of the nutrient assimilation. Water potential plays a key role in solute uptake. Root hairs increase the surface area of the root and also increase the osmosis. If the nutrient potential is more negative within the plant than the surrounding cells, then the nutrients will move from higher solute soil concentration to lower concentration in the plant. There are three physical processes: simple diffusion, facilitated diffusion, and active transport. These three topics are covered extensively in the Plant Physiology Recommended Textbook. I will look at these in some detail here, but please cross-reference the notes that you hear here with your reading in this text. So let us start by looking at passive transport. Simple diffusion is also called passive transport, and you can think of it as the spontaneous downhill movement of molecules. The molecules follow a concentration gradient. They passively move through the lipid bilayer membrane, and no transport proteins or energy are involved in the movement of the molecules. The illustration on the slide shows conceptually how this works. If you look on the left-hand side, you will see a high cluster or concentration of molecules, then a lipid bilayer or cell membrane, and then underneath another cavity, which is the intracellular space, with low concentrations. The next or middle compartment of the image then shows what happens over time is the molecules start to move through from the high concentration to the low concentration. They move through the lipid bilayer. Note there are no proteins or no assistance in this movement. The third part of the illustration on the right hand side then shows that the molecules will reach equilibrium. Once they reach equilibrium, no more molecules will move down, as they are the same concentration above and below. The concepts of diffusion were described by Fick's first law, and this simply says that molecules move from high concentration to low concentration. The exact terminology was the movement of molecules by diffusion always proceeds down a gradient of free energy or chemical potential until equilibrium is reached. When we take this one step further and we look at diffusion, we can begin to understand that diffusion is a process of osmosis. That is, the diffusion of water molecules across a selectively permeable membrane. The net movement of the water molecules through a partially permeable membrane from a solution of high water potential to an area of low water potential. If we go back to the following slide and look at the image, the one part of this definition that was not covered is the membrane itself. The fact that it is selectively permeable means that it will allow some compounds through while not allowing other compounds through. And this is based on chemistry. Now let us look at a different type of movement. This one is facilitated diffusion, and its name does suggest its function. The rapid movement of solutes or ions following a concentration gradient which is facilitated by transport proteins is the definition for facilitated diffusion. Let's see how this works in more detail. The image on the slide summarises the concepts. You will first note the extracellular space, then the cell membrane, and then the space below that is the intracellular space. The nutrients are trying to get, or the plant I should say, is trying to get the nutrients from the extracellular space into the intracellular space. When this occurs by the process of facilitated diffusion, you will have a protein. This protein will be located in the cell membrane and will be called a transport protein. Molecules will go through the transport membranes. Their movement or passageway will be very fast or rapid. This allows for both, one, a very effective movement of solutes, and two, the system is highly selective. And what I mean by that is it will only allow the nutrients through that the plant wants and requires. The following image on the slide shows a number of proteins located in the cell membrane. As you can see from this image, there are many that may be present, and this is in fact quite a complex process. The chemical and physical composition of the solute can be taken up and will determine which protein is used by which solute. In this lecture we will only introduce the concepts here, but in stage 2 of this subject we will explore these interactions in more detail, especially those around nitrogen and sulfur. At this stage, the ideas you will need to take on board is that the process is rapid, and that it occurs along a concentration gradient, and it is facilitated by protein structures in the cell membrane. The very important take-home message is that this type of transport requires a lot of energy. The final process we are going to look at is that of active transport. The definition of active transport is the movement of substances against a gradient of chemical potential, or you can think of this as uphill movement. Where the chemical potential of any solute is defined as the sum of the concentration, electric and hydrostatic potential play a role. Direct use of energy to transport molecule across the membrane. Again, you can refer to the textbook for further details. So let us talk about active transport in a little bit more detail. The first thing to note about active transport is that it usually occurs where you have an extreme concentration gradient. This could be quite typically when the ground is droughting, or when the concentration of nutrients around the root hair, for example, is very low. So the route of nutrient transport, it starts in the extracellular space. It is moved through the cell membrane into the intracellular space. As the name suggests, active means that it needs additional energy. The transport route is best used because of the high energy input when there is a concentration gradient. Concentration gradients can be measured by the amount of sodium and potassium in both the extracellular and intracellular spaces and how they move around. Proteins, which are different from the facilitated proteins, are embedded in the cell membrane. The compound of transport moves through the membrane. The membrane requires a large amount of energy. This energy is in the form of the compound ATP, which is also called adenosine triphosphate. When ATP is used, it changes its chemical composition and becomes ADP, adenosine diphosphate. It is the movement of inorganic phosphate that enables the exchange of energy. Most of the enzymes that perform this type of active transport are the transmembrane ATPases. They require a sodium-potassium pump. They maintain cell potential. Redox or photon light energy may also be used in active transport systems. The slide on the screen, and the one I described previously, describes the sodium-potassium active transport pump. Potassium ions accumulate passively. Active uptake when there is external potassium is low. Sodium activity pumps out of the cytosol into the extracellular space and the vacuole. Thus, excess protons actively pumped from the cytosol. Ions are actively taken up into the cytosol. Calcium actively transported out of the cytoplasm. There are some additional resources which will help you understand the transport mechanisms. Please watch the 3-minute video called Absorption by Route Active and Passive Transports. This can be found at the following link and is available on the Moodle website. The graphics in this video explicitly show how the passive and the active transport mechanisms work. You will be able to take some notes from this video and put them in your lecture notes here. 3 mechanisms have been described for transport of nutrients. These mechanisms are utilised in the uptake of nutrients from the soil matrix to the phloem. Different routes of nutrient uptake can occur. The route will determine which mechanism of uptake is employed by the plant. So let's take our discussion of routes of transport to more detail. If we look specifically, we can separate transport into what we call short distances and long distances. Short distances, as they describe, tend to be 1 or 2 cells across, while long distances tend to encompass the whole plant. Water and minerals can travel through a plant by one of 3 routes, from the tissues to the organs. There is out of one cell, across a cell wall, and into another cell. This type of mechanism is called the transmembrane route. You will see the transmembrane route shown in green on the slide. This figure is a very good figure as it does demonstrate the 3 different routes I'm about to describe. The second kind of route is the symplastic route. This is where, in the slide, you can see the blue line. So the compound will go into one cell. It will not go across the cell wall but travel directly through to the next cell, and so on. The last route is called the apoplast route. This does not go into any of the cells, and in fact it goes into the non-cellular components. The next couple of slides allow us to explore in more detail both the symplastic and the transmembrane routes. So the symplastic route goes through the cytoplasm. The solute enters the root hair cells across a partial permeable membrane by osmosis. The solute moves from a high water potential to a low water potential in the cell. Solute moves across the route from cytoplasm to cytoplasm down the potential gradient. It passes from one cell to the other via the plasmodesmata. Solute moves into the xylem by osmosis. The only way across is the endoplasm, normally the most important pathway and the most common. When you are learning this route, take a moment to review not only the processes, but also the route the compound is travelling. The less common route, but still important, is the ATPase route. In this route, the solute moves through the cellulose cell wall and into the intracellular spaces. The permeable fibres of the cellulose do not resist most solute flow, and hence this route can exist. The solute cannot pass the endodermis by this route. Because the Casparian strip in the endodermis cell wall is impermeable to water, due to the waterproof band of the compound cerberin. So all solutes must pass the endodermis via the cytoplasm. Therefore, we say it is under cellular control. ATPase route is important when transpiration rates are high, as it is faster and requires no energy. The following slide illustrates the lateral transport of minerals and water en route. This is a very useful slide, and I would suggest you taking some time to pause the lecture and reviewing it and understanding the different processes. First of all, it shows you a cross-section through a route. As you can see, all of the compounds, the solutes of interest, will come in through the root hair and eventually end up in the xylem, and this is shown by the image in the middle of the slide. There are two principal routes that this transport can occur: the aplastic route and symplastic route. In this figure, the aplastic route is shown in pink, while the symplastic route is shown in blue. Things to note: the solutes will start from the root hair and always end up in the xylem vessels, where they can be translocated to the rest of the plant. Both routes go through the root hair, the epidermis, the cortex, endodermis, and into the xylem vessels. How they go through these structures, however, is different, and you can follow both the descriptions and the images to see how these are different. The third slide, or the third sub-image here, shows you more detailed of how they go through or bypass the cells. This again helps strengthen the concepts that we are trying to describe here. Please take some time to read each of these stages 1 through to 5. Now that you have understood the main mechanisms and the main routes of how solutes enter plants, there is another level of complexity that we can introduce, which explains in further detail this complicated relationship. Most plants, we have discovered, have mutually beneficial relationships with fungi and/or bacteria, which can facilitate the absorption of water and minerals from the soil. These organisms are usually referred to as the Mycorrhizae, and they tend to be symbiotic structures, which consisting of plant roots which are united with the hyphae from fungi. The image on the slide shows one such relationship. The brown structure in the centre is the root, and the white structure around the outside is the fungi structure. This image shows white mycelium of the fungus around this pine root. What you will note is that the fungus provides a large surface area, therefore you can actively increase the absorption of water and minerals from the soil. Symbiotic means that it is good for both components of this relationship. The plant gets to absorb more water and nutrients, while the fungi is fed or given substances to enable this relationship. There is what we call essential reading associated with this topic. By essential, it means that you need to do the reading and make some notes on it in order to complete your lecture notes. You may be tested on any component of this reading. Please refer to the Tays and Zeiger 2010 Plant Physiology recommended textbook for reading the chapter Mineral Nutrients, Chapter 5. This is based on the 5th edition. If you have an early edition, it may be a different chapter. In this chapter, please concentrate on the section The Roles of Essential Elements and Nutrients Disorders. You will learn about root systems and interactions with soil, mineral root entry and mycorrhizae associations. Another chapter I'd like you to review is chapter 12 from the same textbook. This is on the topic of assimilation of mineral nutrients, selection of water absorption by the root. In this chapter you will learn about nitrogen, sulfur and oxygen assimilation. Please make notes and put your notes with your lecture notes here. I would also like you to watch the following YouTube video. This link can be found on Moodle. This will review much of the mechanisms and transport routes discussed in this lecture. It will help you to understand the processes, but also review and ensure that you've understood what has been covered in this lecture. Any additional notes from this YouTube video can be inserted into your lecture notes here. So let us review what we have learned in lecture 3. We have concentrated on the role of nutrient uptake in roots. We have also touched on the uptake of water. We have learned that there is a number of uptake mechanisms, and this can be quite complex depending on the route the compound takes up. The way a compound is taken up depends on both the physical and the chemical composition of that compound. We've learned about uptake mechanisms such as passive, which also covers diffusion, facilitated and active uptake. We've learned that nutrients can travel 3 different routes from the soil matrix to the transport vessel, the phloem and xylem. That these routes may take on passive, facilitated and active transport. That the root structure is very important in both the uptake mechanism and the root. Nutrient uptake can be aided by fungi and other organisms called mycorrhizae. And we have been introduced to some further resources to complement this lecture, including videos, essential reading, and there will be a quiz available for this topic. This brings us to the end of lecture 3.